Hi, everyone. As usual, I'd like to begin by thanking you for joining us and the Chan Zuckerberg Initiative for funding. We're excited to be joined today by Yasset Perez Riverol from the EBI in the UK, and he will be presenting the NF-Core pgdb pipeline, which is focused on helping users create proteogenomics databases using ENSEMBL as the reference proteome database. And Yasset will be telling us more during the talk. If you have any questions for Yasset, either unmute yourself at the end of the talk or use the chat function, and I will relay the questions over to him. Thanks very much for agreeing to present for us today, Yasset. I'd like to hand over to you now so that you can start your talk.
Okay, thank you for the presentation. I hope this can be seen now. Today I will be talking about proteomics in NF-Core, and specifically one use case is proteogenomics analysis of non-canonical peptides. My name is Yasset Perez Riverol. I am team coordinator of Proteomics services at ENSEMBL EBI. And I think this talk will be more about how it's mainly a use case about how you can use, or we have been using NF-Core and Nextflow for more than a year right now to analyze our data. Rather than the internal, rather than a technical talk about insights of NF-Core, I guess you guys have more talks in the past talking about DSL2 modules and so on, but this will be more about what you can do right now with NF-Core pipelines to do proteogenomics analysis. First, I will, what is mass spec, especially bottom-up proteomics? In summary, you start by extraction of cells, your sample, and you try to separate the protein from that sample, and after some preparation, that especially digestion, you convert that into peptides, and then there is mainly two instruments that give you finally millions of mass spectra. Each spectra corresponds to a peptide, and all the those spectra are a relation between a mass and an intensity for each mass, and these are fragments of that particular peptide correspond to one of each peaks in the mass spectra. And until that, you have let's say, analytical part, the instrument part. But then is when you have bioinformatic tools that need to first taking this result from the spectrum, mass spectrometer, the spectra you need to assign to each of these spectrum peptide, with some sort of a scoring system that is multiple ones. And then when you have a list of peptides, you need to infer the protein that actually is the one that is interested mainly in biology. And finally, you need to do the quantification of the protein, how much of that peptide and that particular protein is in the sample. This is mainly, really in summary, what bottom-up mass spectrometry is. Mainly we'll be talking about this part, what we have done in this part. What is proteogenomics? Proteogenomics is using mass spectrometry to identify peptides and proteins, but using genomics and transcriptomic information. For example, to improve gene annotation, or for example, to do prioritization of gene by using the protein expression information. In the proteogenomics, for example, if you have identified a mutation or a variant, you want to see by mass spectrometry if that particular mutation can be seen or expressed, and how much of that protein is actually present in the sample, the protein expression of that particular protein for that particular mutation. That's what we want to do with proteogenomics. It's actually the bridge between genomics, transcriptomic, and proteomics becoming really popular. There is a really nice review from Alexei Nesvitsky in Nature Methods about what is proteogenomics and what are the challenges in proteogenomics. Then, to perform proteogenomics, the main task is that you need to, after, as I explained before, you have the mass spectrometry data. Most of the peptide sequence identification step, the one that you actually assign the mass spectra to a peptide, use a protein database to do this identification. In normal, let's say, when you work with a normal proteomics experiment or common proteomics experiment, you use UniProt or ENSEMBL to do that. However, when you do proteogenomics, you need to add to this proteogenomics database, you need to add the novel peptide, what we call non-canonical peptides in this case, which is basically a translation from the genomics information, let's say from a mutation, from a variant, from a pseudogene into a peptide information, okay? This can be summarized in this. I mean, you have the canonical peptides from ENSEMBL. This is the reference database from ENSEMBL. And then you need to start adding, for example, alternative or non-canonical peptide, alternative reading frame, pseudogene, non-canonical RNA to that particular database. You need to add also, you can add also if you have variant information from different sources like Cosmic and so on. Or you can add a patient-specific data from the BCF data. You can add that and translate that into a proteomics database. And this is actually what we have done this year. We have developed a tool using NF-Core, and I will explain a little bit about how it's done, to generate the proteogenomics database. Because as you can see, there is multiple steps if you want to build a proteogenomics database. Multiple steps that are involved to finally have a proteomics database, including the last step, which is a decoy generating step, where actually you take the target database or the sequence database, and then you attach to the database a decoy generation for the peptide identification steps to compute FDR score for each peptide. We first, before any development related with Nextflow or NF-Core, we actually developed a tool in Python that does the simple steps, like, for example, performing the translation from gene and transcript into protein databases, for example, for pseudogene, low non-coding RNA, and so on. Also, for example, for common or most common mutation databases like Cosmic or cBiopol to translate that into protein databases. And also simple algorithms inside to compute, for example, decoy, to generate the decoy sequence for large proteogenomics databases, or for example, for removing stop cones and so on. This is really important for proteomics. I will not go into detail, but mainly to process the proteomics database. And then that tool enables to do those independent steps. However, if you want a proteomic genomics database, and this is the diagram of the pgdb, you really need to combine all these steps depending on which type of database the user wants to generate. And if you can see in this graph, this is the DAG of we developed an NF-Core pipeline called pgdb, which basically enable the user to combine all the tasks that provide the Python tool to generate the database that the user wants. In this case, for example, starting from downloading the FASTA file from ENSEMBL for the specific proteome, then there is a way for the user to attach to the database, for example, alternative open reading frames if they want, for example, pseudogenes and so on. These are, as you can see, if for those familiar with Nextflow, these are all conditional steps. Mainly the user, this is controlled by parameters in Nextflow. The user can decide they want to attach to the final database all these steps. There is another big block here where the user can attach also variants and mutations to the database. This is also controlled by, oh, sorry. This is also controlled by, let me see if I can move this here. Okay. This is also controlled by the pilot, but actually one of the cool things is that you don't need to do anything manual. Let's say you don't need to download the data from Cosmic. The pilot allow you to provide a user in Cosmic and also an FTP, and the pylon itself allow you to download the original data and to translate into protein sequence database. And finally, what we call decoy generation database cleaning, which we actually remove the sequence that are not interested, and also generate the final decoy database. And that's what pgdb does. We have tested, of course, the pylon with multiple samples. For that study, we performed a reanalysis of public proteomics data, including 6 proteomics data set, more than 65 cancer cell lines, around 20 million spectra. And we identified more than 5 million canonical peptides. These are the most common ones, the ones that, and almost 300,000 non-canonical, 2,000 mutations, and 21,000 variants. You can see here the non-canonical mutation we have identified by each of the cell line. Interestingly, the pgdb as a pylon also allow you to generate, for example, tissue-specific or cancer cell line-specific databases. And this is only possible because we have a pylon like this that we can filter out for each specific cell line or cancer cell line, which are the mutations in Cosmic that you, or C-viral portal that you want to use. In Pride, we are interested to really map all this information to ENSEMBL coordinates, and we have an independent tool that does that after the reanalysis. And you can see in ENSEMBL for one specific region of the genome, what peptide has been seen and in which kind of cell line or sample condition. Then until here is pgdb and how do we generate the databases, why PG is important for us, and what kind of task it enables for proteogenomics database generation. As you can see, you have multiple tasks, all of them combined, and you can generate Nextflow allow and NF-Core allow us to generate customized databases. You can, for example, generate the reference proteome from ENSEMBL plus only Cosmic variants, or you can attach a part of Cosmic variant. You can also attach, for example, open reading frames, or you can novel open reading frames, or you can attach the of pseudogenes and so on. This is based highly customizable. But also you can add, for example, if you have a BCF from patient data, you can attach also the variants from the genomic side. That's what we do. But how do we analyze the data? I think it's not the goal of this presentation to talk in detail about how you can do mass spec, but I want to basically highlight what a group of people in NF-Core is trying to achieve also for data analysis. How you can go from mass spec and the proteogenomics database until the peptides identification and the protein quantification. And this is quantms, is an all-in-one DSL2 proteomics pilot. Originally, we developed Proteomics LFQ, is released in NF-Core, but it's DSL1. But working with DSL2, one of the features that we saw is that you can really reuse huge part of the pylon where 2 types of analysis are possible. Then that they reuse this part of the pylon. This is the module base, has allowed us to move into one, let's say, looks like a heavy pylon, but more customizable pylon called quantms that allowed us to do TNT, labor-free, and DIA LFQ data analysis. The DIA part is still under development, but the TNT and LFQ is done. Hasn't been released, but it's done. We are now benchmarking this with existing in data. The most prominent feature is that is DSL2 based. allow us to do label-free quantification data analysis and TMT data analysis. I think it's really relevant because this is not common in proteomics. It's based on a standard file format, SDRF, MCML, and MCTAB, and it's based on OpenMS tools and open source tools. This is also the first pipeline, I think, out there that is based on open source tools completely, which is actually not really common either in proteomics. For input and output of this pipeline, we use recently developed by this community, people that have been working in this pipeline also, an SDRF for proteomics, which is a tab delimited sample metadata and experimental design. We actually know that in order to be able to reanalyze data with Nextflow, we really need some kind of input tab delimited that organize the sample data and how the experimental design is done. And by working on this, we actually, with the experience of Nextflow, I would say an NF core, we actually develop our own representation, also with experience with other fields, Array Express and so on. We actually develop a tab delimited file format that when people submit data into Protein Exchange, their data to Protein Exchange, they need to express this into a tab delimited file format that will enable us in the future to reanalyze the data. The second input file is MCML, which contains the raw data, and we have a step for some instrument to convert to MCML if it's possible. And the output at MCTAB, an additional tab delimited file format to help the downstream data analysis. This is completely new in proteomics. Nothing in proteomics has been done basic on, at least in workflow manner, basic completely in standard file format from the input to the output. There is a common pipeline for peptide and protein identification for TMT and label-free. I will not go into details, but mainly it used two search engines, Comet and MSCF+. It used Percolator to boost the identification of both search engines. Then they combine both search engines using the consensus ID tool, and then finally perform for localization analysis using Lucifer. After that, we have, depending, and this is where both branch split, if the user is using an LFQ pipeline, a library-free pipeline, a tool called Proteomics LFQ will perform protein quantification, inference, feature detection, and match between runs. If the user is using a TMT, we will be using isobaric analyzer to perform quantification between TMT channels. Most important, or it's quite important, that all this data is exported into MCTAG, which is a standard file format for proteomics. And we have tested already that the quantms pipeline actually can export directly into Protein Exchange, meaning that when you finalize your experiment, you can perform a complete submission into the main database in Proteomics, which is private, but also in Protein Exchange, because it's working with a standard file format. And then people can download the data, visualize it, and so on. One problem that we have actually in proteomics is that we don't have something like multi-QC. We don't have something that enables us to visualize the QC reports of a proteomics experiment. We have worked in a tool, it's called pMultiQC. I don't know, I haven't talked with Phil about branding, but I hope this is fine. That is actually an extension of MultiQC, but it's mainly working for proteomics. And you can actually have really nice plots like this when you see the number of picks per MS, the number of peptides identification by each search engine, and the final result by each of the MS run. You can see one example here. This is a really active development. We have a really nice feature that enabled to search peptides identify in the web by using SQLite in the backend hosted by the HTML. This is quite nice and powerful tool. We are doing quite a lot of work there. As anyone knows, in NF Core, you can see also all the reports of the pipeline. We have analyzed a lot of data set with NF quantms, and you can see all the steps here, the CPU usage and also memory usage. This is more for people that is actually now arriving for the first time to NF Core and want to see what kind of reports you can see when you have your final reports. For those ones that know NF Core, they know that this is one of the great features of having the pipeline in your pipeline in NF Core. The DIA part is under construction, update construction, but the TNT and neighbor-free are done. I want to thank to 3 teams that have been actively working in this pipeline. First is the OpenMS team, Juliano Santimo. They have been working on every tool that works within the pipeline. Our team that actually have been working in both pipelines, and from the Karolinka team, Hussein has been worked with me in the development of the pgdb and also the reanalysis of the cancer cell line data sets. Thanks for the opportunity and questions.